So far, we talked about the determinant systems in metabolic modeling approach. So we solved the set of equations such that the degrees of freedom is made zero by specifying some of the unknown rates. But as I said, metabolic networks usually have many more unknowns than the number of equations, number of intracellular metabolites. So our generic approach in mass balance based metabolic modeling will be flux balance analysis. So we will have underdetermined systems, and most of the time we will use optimization to predict rates. We have already defined underdetermined systems before, right? Degrees of freedom is greater than zero. In the solution of such system, three different constraints are used. One constraint is S.V equals zero, our mass balances, right? We can refer to this as mass balance constraints or equality constraints. The other constraint is the measurement constraints. So we are familiar with this constraint too. So for some of the rates, let's say V5, we know its value. Let's say its value is 6. So this is also a constraint, right? Our solution must satisfy this. V5 must be 0. Sorry, V5 must be 6. So you introduce a constraint to the problem of solving unknown rates. The third constraint, we didn't talk about it so far in the determined systems part, is the reversibility constraints. There are two alternatives for a reaction. The reaction can be either reversible or irreversible. Irreversible means that if you write the reaction like this reaction only proceeds from A to B. It cannot proceed from B to A. So the reaction is irreversible. Reversible means the reaction can proceed in both directions. Either B can be formed from A or A can be formed from B. If the rate of a reaction is predicted as -5, for example, this means that the rate is 5 and the reaction proceeds in reverse direction. So from B to A is the real direction of this reaction. So minus in the reaction rate means the reaction proceeds in the reverse direction compared to the direction you considered it initially. So the reversibility information also provides constraints for our problem. How? We can easily say that irreversible reactions, they can never take negative values. So irreversible reactions will always take values between 0 and plus infinity. So they will always be positive. You constrain them to be always positive. Reversible reactions, on the other hand, they can take any values between minus infinity and plus infinity. So, in flux balance analysis formulation, You need to define your mass balance-based constraints, your equality constraints, your measurement constraints, so some of the rates will be defined as measured. And you should also define the ranges for the reaction rates. For reversible reactions, the range will be between -infinity to +infinity. For irreversible reactions, the reaction rate can take only values between 0 and +infinity. These are the three constraints used to define the problem for underdetermined systems of metabolic networks. So basically you have a set of constraints and you seek to find that satisfies all those three constraints. That's why flux balance analysis and similar approaches are also referred in literature as constraint-based analysis. You use mass balance constraint, you use measured rate constraints, you use reversibility information as constraints, and based on those constraints, you try to predict rates using, for example, optimization. So flux balance analysis, or FBA, is the most common constraint-based analysis method. There are other approaches. There are some approaches that don't use optimization. We will talk about those in the coming weeks. But the most commonly used approach is flux balance analysis, and it is a constraint-based analysis method. It uses optimization to calculate unknown rates. Optimization requires an objective function to be optimized. So you should get familiar to the concept of objective function. Before proceeding, I would like to share with you the first FBA example in literature. It was published in 1986 by the scientists from Oxford Polytechnic. So the title says, Fat Synthesis in Adipose Tissue, an Examination of Stoichiometric Constraints. Compared to the other examples we have seen so far, the focus here is not microorganisms, but mammalian systems, adipose tissue, fat synthesis. So in that sense, this work is unique. And it has a very tiny abstract. Let's check the abstract together. So it talks about net balance of synthesis, degradation, and transport for all intermediates in the pathways. So remember, we write balances around intermediate metabolites. That's what it talks about here. And starting from glucose, there are a lot of reactions that lead to fat synthesis. So it also talks about the balance of fluxes. And it says that linear programming was used to examine the interactions between these constraints. You see, the term constraint-based analysis was coined in 2005-6 maybe, but more than almost 20 years before, David Fell and Rankin Small, they used those terms, constraints and linear programming, etc., in their paper. And they tested the case of excessive ATP synthesis. Let me share a figure from their paper. So they share in this figure relative fluxes they have predicted by using the objective function of maximum economy of glucose utilization during fat synthesis. So there is this extracellular environment. So this is the system boundary. Glucose is coming inside to the cytoplasm. And this is the metabolic network they have considered. So this is mitochondrial membrane. Malate, pyruvate, ATP passes through the mitochondrial membrane. Here we see some reactions of TCA cycle, citrate formation, oxaloacetate, malate, alpha-ketoglutarate, etc. And here you see oxidative phosphorylation, the use of NADH and FADH to make ATP. This is the glycolysis. This is the pentose phosphate pathway. This is the ATP for maintenance. And here you see fatty acid synthesis pathway. So this system is underdetermined when you check the degrees of freedom. And you need optimization to predict rates. Remember that underdetermined means there are many solutions to satisfy the given constraints. And among those many solutions, we choose a specific one which corresponds to the optimum value of one of the unknowns or a group of unknowns. So objective function is the function that tells us which unknown to optimize. In this case, as far as I remember, they fixed the fat production rate to 1, and they have minimized the consumption of glucose here. So their objective function was glucose consumption rate, minimization of glucose consumption rate. Objective function can either be minimization or maximization. You try to optimize, you try to find the optimum value of an unknown. This optimum can be the minimum value or maximum value. And in this case, they wanted to identify the minimum amount of glucose consumption rate to achieve a production rate of 1 unit of fat molecule. And they used linear programming, and they were able to predict all the rates that corresponds to the minimum consumption rate of glucose. So in this specific condition where glucose is used at the minimum level to achieve 1 unit of fat synthesis, these are the predicted rates. So this example, this paper is the first example in literature that uses linear programming optimization for an underdetermined metabolic network system and predicts the rates. So let's talk more about flux balance analysis, the solution of underdetermined systems by using optimization. Here I will cover a very simple FBA example. So this is the only thing given to us. So we have said that this equation is given. This is like our mass balance equation. So equality constraint. So based on this, we have one equations and two unknowns, V1 and V2. So degrees of freedom is 1. For this ultra simple system, we have an underdetermined system. And we are given that V2 is irreversible. So if we write the reversibility constraints, V1 is between plus infinity and minus infinity, because it's reversible. It can take negative values. And V2 is between 0 and plus infinity. So the question is, given those constraints, the mass balance constraint and the reversibility constraint, what is the minimum possible value of V1? So this means that the objective function in solving this problem can be minimization of V1. We have one equation, two unknowns. So we have many solutions that can satisfy this equation. For example, V1 can be 4, V2 can be 1, which will make 3. V1 can be 1000, V2 can be 997, and you can write many other alternatives, right? But among all those alternatives, in this problem, the question is, what is the minimum possible value of V1? Here, the value of V1 is 4. Here, the value of V1 is 1000. So what is the minimum possible value of V1? For example, if V1 is, can V1 be minus 1000? In this case, V2 has to be plus 1003, right? Then, sorry, it has to be minus 1003. So minus 1000 minus 1003 will make 3. But does this violate any of our constraints? The answer is yes, because V2 has to be irreversible. So this is not possible because here V2 has a negative value, right? So this violates one of our constraints. So let's focus on the question again. What is the minimum possible value of V1? We know that V2 can have any value between 0 and infinity. Previously, I have forgotten to put those equal signs, but they can also be 0, of course. So the minimum value of V2 is 0, and maximum value of V2 is infinity. And we want the minimum value of V1. So I said that V1 can be 4. In this case, V2 will be 1. It will be 3. V1 can be 8. In this case, V2 will be 5. The minimum value will be 3. So in terms of minimum possible value, this is more minimum, I mean smaller than this one, right? Can this be smaller? Yes. So this minimum value of V2 is 0. Here V2 was 5. Here V2 was 1. So if V2 has its minimum value, which is minus 0, what can be the value of V1? The answer is 3, right? So we have solved an optimization problem without using any computational algorithm, right? So by using the constraints of mass balance constraints and also the reversibility constraints. And by using the objective function, among many possible solutions, we choose one which corresponds to a specific objective function. So the solution to this problem is 3 to 0 for the given objective function. So this is how, among many other solutions, many possible solutions, we choose a specific one that corresponds to an optimization criteria, that corresponds to a given objective function. Now I am talking about simplistic objective functions, right? There's only minimum value of V1 or maximum value of V2. Of course, there can be more complex objective functions. Now I will talk about objective functions, and that will be more clear for you. Optimization in general can be grouped into two: convex optimization and non-convex optimization. Our focus in this course, if you go to literature, 99% of the research on metabolic network modeling is for the convex optimization. So linear programming or quadratic programming is types of convex optimization. If you have an optimization problem, you need an objective function. We already talked about this. So objective function tells us what we want to optimize. So what we want to minimize or what we want to maximize. Here you see example objective functions. So objective function can be very simple, minimization of V1, or it can be more complex, minimization of the difference between V1 minus 2V5, or maximization of the third power of V7 minus square root of V6. So objective function can be very complex, but most of the time for biological systems, for metabolic networks, our objective function will be very simple, and we will use straightforward optimization algorithms such as linear programming or quadratic programming to solve for the unknown rates. So linear programming means the unknowns in the objective functions are linearly combined. So objective function is a function of unknowns. So it's a function of unknowns. So unknowns in our case are rates. So objective function is a function of rates in our case. It can be a single unknown or it can be multiple unknowns, as I said. For linear programming, these unknowns has to be combined linearly, which means that there shouldn't be any power or square root, of the unknowns. So in this case, you see the power of all unknowns, so they are linearly combined. In this case, we can apply linear programming. If there are terms in our objective functions that includes squares, quadratic terms, then our objective function is quadratic because it includes both power 2 and power 1 terms. And in this case, we need to use quadratic programming. So let me repeat. In metabolic network modeling, our focus is convex optimization, and we will mostly use linear programming or quadratic programming. The objective function is a function of a function of unknowns. If those unknowns are combined linearly, we use linear programming. But if there are any square terms, then we use quadratic programming. Those cases are more advanced cases, and almost never we need such. We will have such objective functions, so that's out of scope of our focus in this course. And when you go to literature, as I said, in 95% of the case, you will only see linear programming and quadratic programming cases. So objective function will be a linear function or a quadratic function. And this linear programming is much more common. So, then the question is, how will we choose our objective function? There are many unknowns. Which one will we maximize, or which one will we minimize? Will we maximize only one of them? Will we maximize the sum of 5 of the unknowns? Will we maximize the difference of 2 unknowns? There are many alternatives. The answer is, the chosen objective function must be biologically meaningful. So this is the requirement. You cannot randomly choose an objective function. It must be in accordance with the phenotypic behavior of the organism of interest. And the most commonly used objective function in metabolic network modeling in constraint-based analysis approach, in FBA, flux balance analysis, is the maximization of biomass reaction. If the organism is a microorganism, I think 95% of the papers in literature use biomass maximization. And since biomass is just a single reaction, right, V biomass, you maximize this. So your objective function is linear, right? Just it includes a single term, and this term doesn't have any square or something. So that's why most of the works in literature are based on linear programming, because they have this very simple objective function, which is a linear objective function. So why is maximization of biomass reaction commonly used in literature? Is it biologically meaningful? Is it in accordance with the phenotypic behavior of the organism? Maximization of biomass reaction means maximization of growth, right? Biomass reaction defines the growth of the organism because it defines the production of macromolecules, lipids, carbohydrates, nucleic acids, and proteins. When the organism grows, right, divides, for example, the microorganisms, they divide to make new cells. So this means more and more production of those macromolecules. So microorganisms try to grow as much as possible. That's the assumption made when you choose maximization of biomass as the objective function. You assume that microorganisms try to grow as much as possible. And we know this, right? E. coli, Escherichia coli, when you grow it in an environment, if you observe it with a microscope, they will grow in a very fast rate. The same for many other microorganisms. So, the first example of FBA in 1986 was for adipose tissue, so it wasn't microorganisms. They used a different objective function, minimization of glucose consumption. But in 90s, the examples with microorganisms started to appear in the, and they used always maximization of biomass reaction. And they were able to show that by using this objective function, for example, so they predict the rate of biomass reaction, they compare it with the measured growth rate, and they are in agreement. For example, they predicted by using this objective function the rate of ethanol production, and when compared with the measured ethanol production rate, they were in agreement. So in a way, they have validated this choice. Later, in 2002, a more direct experimental proof of this objective function of biomass reaction rate maximization was made available by Paulson's group. The paper was published in Nature. So this is what they did. Normally, Escherichia coli and all microorganisms, they grow on glucose. Glucose is the natural substrate for microorganisms. They use glycerol to grow Escherichia coli. So glycerol is not an optimum substrate for E. coli. Don't focus too much on the lines in this graph, but just I won't go into details not to make it complicated. This line, red line, shows the optimum line. Okay, optimum line calculated based on flux balance analysis. And each dot shows the growth rate of the organism for a given oxygen uptake rate and glucose uptake rate. For this specific glucose uptake rate and oxygen uptake rate, the growth rate of E. coli on glycerol was this. So they have replicate experiments, not only one. So for this experiment, this glucose uptake rate, this was the oxygen uptake rate, and this is the corresponding. So this red line, as I said, corresponds to optimum growth calculated based on flux balance analysis. This is day 0. So when they measured growth rates from day 0 samples of E. coli grown on glycerol, their replicates, most of them were away from the optimum line. They kept growing the organisms on glycerol, so they continued the experiment, and at day 60, all the replicates had the growth rate which was very perfectly predicted by flux balance analysis. So the message of this paper is this. Maybe this wasn't an optimum carbon, optimum substrate for the organism, but when the organism got used to this substrate over days, at the end, it started to use this substrate in a most optimum way for its maximum growth rate. So this is a validation of the use of growth rate maximization or biomass reaction rate maximization when using flux balance analysis. I will shortly talk about how we can make our problem ready to be solved by using optimization. You can do this in MATLAB, you can do this in R. For now, I will talk about Leanpro function in MATLAB. So this function is available in the optimization toolbox of MATLAB. And it requires a set of inputs. Now we will talk about which of those inputs correspond to the mass balance constraints, which of them corresponds to the measurement or reversibility constraints. So Leanpro function in MATLAB was designed to solve problems constrained by this set of equations. So here x is unknown, okay? X is the unknown to be calculated, so it corresponds to our reaction rate vector, our flux vector. So A, EQ and A are the matrices. So which of those two forms resembles our mass balance constraints? The second one, right? Our set of equations is S dot V equals 0. So we have equality-based constraints. So this is the form that we have. We don't have any equation based on matrices, based on balances, and includes inequality. So we don't have this in metabolic network-based modeling approach. So our A and B will be empty vectors or empty matrices. And this AEQ is our S matrix. And this BEQ is the right-hand side of the equation. In our case, it is zeros. M by 1 vector of zeros. Right? S is n by m by n. M rows, n columns. V is n by 1. We have n unknowns. So the resulting vector, here called BEQ, will be m by 1. So m from here, 1 from. So if we want to use Leanpro, we will define empty matrices for or vectors for A and B. AEQ will be our stoichiometric matrix. B will be the zero vector in S dot V equals 0. So the remaining inputs we didn't define so far is F, LB, and UB. And so far we have only defined our equality constraint, our mass balance constraint. We didn't talk about how to define reversibility constraints or measurement constraints. We talked about this already. So let's talk about LB and UB. LB is a vector of lower bounds and UB is a vector of upper bounds for unknowns. So if we have reaction 1 between 0 and infinity, reaction 2 between minus infinity and plus infinity, and reaction 3 again between 0 and infinity. And let's say this is a very simple system with only 3 reactions. Then lower bound of reaction 1 is 0. Lower bound of reaction 2 is minus infinity. Lower bound of reaction 3 is 0. So these lower bound and upper bound vectors are used to define reversibility of reactions, to define reversibility constraints. So in this case, this will be our lower bound vector. So lower bound vector will be n by 1 in size, because we have n unknowns. For each unknown, we will have a minimum value and a maximum value based on reaction reversibility. In this case, we have 3 reactions, so it will be 3 by 1. And it will be 0 minus infinity 0. This similarly, upper bound vector, in this case, it is always plus infinity. It will have, again, n by 1 dimension. And all the elements in the upper bound vector will be plus infinity. So LB and UB inputs here in Leanpro function are used to define lower bounds and upper bounds of reactions based on the reversibility information. What if we have a measured constraint? Let's say, yes, R2 is reversible, but we will use the measured value of R2, which is 5, as input in our analysis. So we want to specify this specific value. How would we define it? The measurement constraints are again defined by LB UBs. How? So this was our LB vector, right? So reversibility constraint says that R2 can take any value between minus infinity and plus infinity. Okay, it can take any value, but I already know which value it took for this specific case, which is 5. So in LB vector, rather than minus infinity, I should write 5 for R2. Its minimum value is 5. And its maximum value is not plus infinity, but its maximum value is 5 too. So this is how we define measurement constraints. We the same LB and UB values, which is the measured value indeed, for that reaction to define the measurement constraints. So our LB vector in this case will be 3 by 1. So the elements will be 0, 5, 0. And the upper bound vector will be plus infinity, 5, plus infinity. So we talked about all those. I will finish by the definition of F. What did we not define so far? The objective function, right? How will we define objective function? In this case, it is linear programming. So I know that all unknowns, and the objective function will be a function of unknowns, and those unknowns will not have any power other than 1. So F is used to define objective function. Remember, objective function is a function of unknowns. So what will be the definition of F vector? It will be in parallel with the number of unknowns, right? I have n unknowns, so F will be n by 1. F will be n by 1. Let's say in this 3 reaction example, let's say our objective function is maximization of V3. So objective function F is only a function of V3. There is no V1 in objective function. There is no V2 in objective function. And V3 is in my objective function with a coefficient of +1. So this is how I define f. F will have the size of unknowns, and this objective function is defined in a vector format. So there is no v1 in the objective function, so the first element will be 0. There is no v2 in the objective function, the second element will be 0. But the third unknown, v3, is in the objective function with a coefficient of +1. So I write it here, like this. Just a final example, and I will finish. Let's say your objective function is maximization of 2v1 minus 0.5 v3. And in this case, your f vector will be 2, the coefficient of v1, no v2 in the objective function, and the coefficient of v3. It's again n by 1, 3 by 1.